B cells are lymphocytes that are produced in the bone marrow of mammals. Each B cell has a unique receptor protein on its surface that will bind to one particular antigen. Once a B cell encounters its cognate antigen and receives an additional signal from a T helper cell, it activates. Since the focus is on phagocytosis, we will focus on plasma B cells. These are large B cells that produce large amounts of antibodies, which assist in the destruction of microbes by binding to them and making them an easier target for phagocytes. In this example, Jake represents a foreign substance that has alerted B cells to release antibodies. The red ball in Jake's hands represents the antibodies that have attached themselves to the antigens on Jake. Each tip of the Y of an antibody contains a paratope that is specific for one particular epitope on an antigen. The paratope and the epitope bind together, allowing the antibodies to stick to the foreign substance and tag it for attack by other parts of the immune system. The phagocyte is chemically attracted to the foreign substance through chemotaxis. The yellow ball represents a phagocyte. The phagocyte's plasma membrane then attaches to the surface of the microorganism or other foreign material. Adherence is facilitated by the attachment of pathogen-associated molecular patterns, or PAMPs, of microbes to receptors, such as toll-like receptors or TLRs on the surface of phagocytes. The binding of PAMPs to TLRs not only initiates phagocytosis, but also induces the phagocyte to release specific cytokines that recruit additional phagocytes. During the process of ingestion, the plasma membrane of the phagocyte extends projections called pseudopods that engulf the microorganism. Once the microorganism is surrounded, the pseudopods meet and fuse, surrounding the microorganism with a sac called a phagosome, or phagocytic vesicle. In this example, a blanket is used to represent the phagosome. The membrane of a phagosome has enzymes that pump protons, H positive, into the phagosome, reducing the pH to about 4. At this pH, hydrolytic enzymes are activated. In this example, the second blanket is used to represent a lysosome. On contact, the phagosome and lysosome membranes fuse to form a single larger structure called a phagolysosome. The contents of the phagolysosome, brought in by ingestion, are digested in the phagolysosome by lysosomal enzymes. Enzymes such as lysosomes hydrolyze peptidoglycan in bacterial cell walls. A variety of other enzymes, such as lipases, proteases, ribonuclease, and deoxyribonuclease, hydrolyze other macromolecular components of microorganisms. Lysosomes also contain enzymes that can produce toxic oxygen products, such as superoxide radical, hydrogen peroxide, nitric oxide, singlet oxygen, and hydroxyl radical. Toxic oxygen products are produced by a process called an oxidative burst. Other enzymes can make use of these toxic oxygen products in killing ingested microorganisms. After enzymes have digested the contents of the phagolysosome brought into the cell by ingestion, the phagolysosome contains indigestible material and is called a residual body. This residual body then moves towards the cell boundary and discharges its waste outside the cell. To quickly recap, B cells release antibodies that bind to foreign substances and tag them for phagocytes. Phagocytes are then chemically attracted to the foreign substance through chemotaxis and adhere to the foreign substance surface through the attachment of the phagocyte's plasma membrane. The phagocyte extends pseudopods to engulf the foreign substance, and then fuse to create a phagosome. Then the phagosome and lysosome membranes fuse to form a phagolysosome. Enzymes then digest the foreign substance. Anything left after digestion is called the residual body, which is then discharged outside the cell.